So now let's get back to the term I've been using, chirality, achiral, enantiomers. These are all terms that you've learned before, and so it's time to revisit them. Biologically active amino acids and other molecules are only one enantiomer, and so it's important that we understand what that means. And so if we look at this image right here, we're looking at this dude looking into this analyzer, and inside the solution is a solution of a standardized quantity of, for example, an amino acid that's chiral, any kind of optically active substance. And this whole setup is actually what tells you that it's optically active. Okay, so there's a light source and then there's a fixed polarizer, and that only allows wavelengths of a certain plane of polarization to go through. It's kind of like, well, it's very much like polarized sunglasses. Okay, so only certain plane will go through, and if you just look at this orientation of it, as it goes through the solution of your molecule, the light interacts with the molecules and changes the plane of light, changes the plane of polarization. So if you see that it's going in this way and it's coming out this way, what this dude over here is doing is he is rotating this polarizer until it will allow this light to go through. So this light is actually, this up and down is not correct for the light coming through here, right? And so he would need to change this so that now the plane of those lines right here is going through here for it to come out. So that these lines going up and down, it would be incorrect. That's how it would start out, but then he would rotate this whole thing so that these lines would then start moving. So, and then you get a value in terms of degrees of optical rotation. And so, and then if it's this way versus that way, it tells you whether it's positive or negative. We're not going to get into positive and negative, and the thing to kind of keep in mind about this is that you can't, looking at a molecule, looking at the structure of a molecule, you cannot predict which way, if it's going to be plus or minus, or the angle. Okay, so it is empirical. These are empirically derived values. So anyway, it's useful though, because once you have a table of values, you can identify substances from this, and you can determine if the molecule is chiral, for example, based on whether or not it interacts with light. And so this example right here from the book shows you a chiral carbon in that it's got 4 different substituents, and this is the mirror plane. So you've seen this before, right? But let's go ahead and also look at it this way. And so what I'm doing is I am showing you 2 molecules that are in the same orientation as what we see on the screen. And so I'm holding the hydrogens. Coming out towards you are the blue bromine molecules. The fluorine are the orange ones that are more towards me, and then the green ones are the chlorine, right? And so we see that they're mirror images, and I can do this, and they're mirror images. I can do them anyway, and they're mirror images. But let me put them back here, and the key thing is that we can't just say, okay, well, they're mirror images, but we can put them on the, right? You can't. So if we match up, for example, the green and the orange right there, right? Well, then the hydrogen and the bromine are different. And so that's what it means to be mirror image and enantiomers. Okay, so that's what we have. They are not superimposable. And so gloves are examples that are often used. There's a lot of examples, but let's just stick to molecules for now. So if we look at this molecule right here, this is glyceraldehyde. And so we see that it is a chiral carbon, and that is that it's got 4 different substituents in it. And these are the geometric formulas, and so again, the hydrogen and the hydroxyl are coming at us, and then the aldehyde and the CH2OH are going back into the plane. And so this is a mirror image, and so this down here, this right here is the Fischer projection. And the Fischer projection is just a way to standardize this when you're drawing molecules, so that if somebody says these are the Fischer projections, you know exactly how to interpret that. Okay, and this is important. So let's look at an example here. So Fischer projections, the horizontal lines correspond to the substituents that are coming at you. The vertical lines correspond to the substituents that are going away from you. Okay, so pretty simple, right? And so when we look at this then, when we look at this is an example of L-glyceraldehyde, that's this, right? And this is D-glyceraldehyde. So we're essentially looking now at a couple of different nomenclature mechanisms. And so what I want to bring up right now is that glyceraldehyde is used as the basis for looking at relative configurations in how we're about to discuss this. And so if we look at glyceraldehyde, that is the basis for this Fischer projection, and compare it to an amino acid. And so if you're going to do this with amino acids, which you will, you would then see how the hydrogen is to the right coming at you, hydrogen is to the right coming at you, and the carboxyl group is going back into the plane of the board or into the screen. We can look at determining whether it is L or S. Okay, and so what this is showing us here is that you rotate this so that the hydrogen is in the back. And let's go ahead and look at this from a ChemDraw perspective. Okay, so now let's compare these structures to a three-dimensional model in ChemDraw. So what we're seeing is that this is laid out in the same orientation that this is, and that is that the hydrogen that's coming at you and the hydroxyl that's coming at you is right here with the hydrogen and right here with the hydroxyl group. Okay, and so back we see the C double bond O and the H, so that's the aldehyde group, and then we've got the CH2OH. So the first thing we do is we rotate this so that we tuck that hydrogen back behind that. So that's what we're seeing there. Okay, and so what we need to do is we need to look at the substituents on this chiral carbon. So we've got an oxygen, we've got a carbon, and we've got a carbon. So we know that oxygen has higher priority than carbon, so that is the W position. But since both of these carbons are the same, we need to go ahead and look at what it's attached to. So this carbon is attached to a hydrogen, and an oxygen. And this carbon is attached to a hydrogen and a double bonded to an oxygen. So they don't have any different atoms, so they have the same atoms as each other. Let me go ahead and tuck that back. And so then what do? Well, you can look at this as this essentially has two oxygen because of the bonds. This has one, at least for the purpose of this discussion. So this has a higher priority with two oxygens than one oxygen. Okay, so from that perspective, we then say W, X, Y, and so then we go counterclockwise, just like we see here, and so that is S-glyceraldehyde. So let's compare this to this structure right here. And so let me go ahead and get it oriented. What am I doing? I'm looking for the chiral carbon. So this carbon has two hydrogens, so that's not chiral. This does not have four different substituents, so it must be this one. And so what I need to do then is I need to orient this molecule, if I can, there we go, so that we essentially have it set up so that it's like this. We've got the hydrogen on the right, and we've got the other substituent on the left. And so we go ahead and rotate this molecule so that we tuck that hydrogen behind there, right? And so now we do the same thing. So we know that the hydroxyl takes the W position because the oxygen group is higher priority than either carbon. And just like we did before, the carbonyl group has a higher priority over the CH2OH. So this is W, X, Y, and so since that's going this direction right here, clockwise, we know that that is in the other configuration, and so that's in the R configuration, or we would call that D-glyceraldehyde. And so when we look at these, this is the way to do this, so be able to work your way around this. So let's do this now with a generic amino acid where we're just using R for R group. And the reason we can do that, and even though, right, because you may be thinking, but we're going to prioritize the substituents with the atomic number. And if we don't know the R group, how are we going to do that? Well, for one, glycine, we're not going to do this with glycine because that's got a hydrogen. And so two hydrogens means that alpha carbon is not asymmetric, right? It's not chiral. And so in this particular example, all the R groups is going to be a carbon attached to a hydrogen. And so let's see how that works here. So if we just look at this, let's go ahead and push that low priority hydrogen to the back, right? So it's not obliterated by this, but you can see that it's going in the back, right? And now let's look at the priorities here. And so we know that this is carbon, that's going to be 12. This is carbon, that's going to be 12. And amino group, the nitrogen is 14. So this is priority. This is number 1, okay? And so then what's the difference between this and this? Well, this carbon group has 2 oxygens bound to it. And so that is going to be any of the R groups. And so when we do this, then we are seeing we're going counterclockwise. And so this is the S form, right? So the L isomer. As opposed to on the right side, with the hydrogen in the back, this particular molecule, we start with the nitrogen, and then we go to the C double bonded C with 2 oxygens on it, and then the R group. And so this one is clockwise, so we know it's R, and it's the D isomer. It is that simple. Don't complicate it, okay? And so that's how you can look at it and determine whether it's the L or the R isomer, okay? So I'm forgetting something. The corn crib. So some people like to use this in terms of it's corn for the carboxyl group, CO, R, and then N. Carboxyl, R, and then N. So what they're saying is that when you're moving the molecule around, make sure that you can in one direction do this. In other words, all that's really saying is that the hydrogen is in the back, okay? But sometimes that's helpful if you're not looking at a 3-dimensional molecule. So chirality, we will talk a lot about enzymes and why it matters there. But these are 2 really good examples in your book, although they don't go into it much, and that is ibuprofen, for example. For ibuprofen, only the S enantiomer is anti-inflammatory. That is the one that inhibits the cyclooxygenase, or the COX. So you may have heard of COX inhibitors, so ibuprofen is one of them, okay? And so when we look at this, which of these carbons is the chiral carbon, right? Which of these carbons has 4 different groups? I will leave that up to you, okay? But so basically only one of the enantiomers actually is an inhibitor, and it turns out that when you take ibuprofen, it is a racemic mixture. Racemic mixture means 50-50% of the 2 enantiomers, okay? And so if we find a racemic mixture of amino acids, say, in ice, on Pluto, for example, or on the moon, 50-50, we wouldn't think that it was made by any kind of biology, because at least the biology on our planet favors one kind, and that in this case the L amino acids, right? Doesn't mean that it has to be that on other planets if there are other amino acids, but basically once you have one, evolution on this planet meant that it was one. So we can find racemic mixtures of amino acids that are made chemically without any biology going into it at all. Okay, so if we see that there's a dominant form of an enantiomeric mixture of amino acid on another planet, that would give us an indication that perhaps it was formed by some type of biology. So ibuprofen, when you take a racemic mixture of that, only the one enantiomer works, but it turns out that as you use up the enantiomer, the one that doesn't work actually gets converted to the one that will. So your biochemistry will convert it to the more active form to some degree, and it's pretty slow. And now if we look at this thalidomide, some of you, I'm sure, have heard of thalidomide. So thalidomide was first made in the 50s, and it was used as a mild sedative and used for nausea, and so it worked really, really well. It turns out, though, however, that the other enantiomer, right, the one that doesn't cause sedation, the one that doesn't combat nausea, is a teratogen. So that means that it will form birth defects. Over 4 years, there were over 10,000 babies that were born with phocomelia. And what that means is that they have malformed limbs and other disfigurations. So if you do a search for thalidomide or thalidomide babies, you will see what I'm talking about there. And so when you look at thalidomide then, which one of these carbons is the asymmetric carbon? Which one has 4 substituents on it? So chirality matters, and there's a whole burgeoning area of chemistry called chiral separation. However, with thalidomide, you still wouldn't just say, well, let's use the enantiomer that, you know, is a good sedative and is anti-nausea and doesn't cause birth defects, because in this example as well, your body will convert that molecule into a racemic mixture. So it will form the teratogenic enantiomer. So it's not just as simple like that. However, the upshot, if there is an upshot, because 10,000 babies and then even more problems other than that, is that by understanding this, a lot of chemistry went into understanding what happened, is that structural analogs of thalidomide were synthesized and are used. And so once you know the structure of the active molecule, so long as you can you know, create a compound that does the same thing and doesn't have alter effects, then you're good to go. So again, checkpoints, the ones that are assigned, understand them well, work on this. This is helping you learn, okay? So, and the 20 standard amino acids are limited. Here's a few modified. There's actually many, many more modified amino acids that we found. As part of my PhD, actually, I found a new modified amino acid. So that was pretty cool. Okay, it was very cool at the time. So if we just look at these, I'm really not going to hold you accountable for much right now. When we get into the specifics and how they work, then I'll hold you accountable. But you can see the blue parts are the parts that are different. The one I do want you to know now is this chromophore. So if you see this serine, tyrosine, and glycine together, they will cyclize. So if you can see that this then binds to this. And what happens is these electrons are delocalized in this tyrosine ring, but now, due to the structure of this, the electrons are delocalized over all of this, okay? And a physical manifestation of that is that it will fluoresce when given certain types of light. And next semester in lab, we may work with green fluorescent protein. It's really cool. So anyway, modified amino acids, you can think of amino acids and proteins as the end-all in some ways, but in other ways, there's a lot of little modifications that occur on a lot of proteins to change the chemical reactivity, to change the physical properties to make it fluoresce as a signaling molecule. Is it phosphorylated or dephosphorylated? And that you learned in Biology 208 or 190, whatever molecular and cell course you took before. And here's an example of more derivatives. These over here are neurotransmitters. Some of these are derivatives. This is straight up. Thyroxine, that is a hormone. And then histamine is an inflammatory mediator that's produced by your immune system. So amino acids, there's the basics, but then all of the different amino acids that are formed and the biological activities that they have are wondrous, like all of this. So I think that wraps up our amino acid section for this, and know that we will be building on this.